challenge.
Yes, okay. So can you see my screen?
Yes.
Okay. So I will very briefly give you an overview of the assembly results of the second challenge. As Alice mentioned already, the data sets are simulated using the CAMI sim pipeline, which has been published, which generates all the different data sets you can use for benchmarking. Just to give you an idea of how this works in general, just move something. Okay, sorry, it's hiding my window here. Okay, so we start with an initial assembly of a real genome, of an isolate genome. From this initial assembly, we simulate reads, and we simulate short reads and long reads, as Alice mentioned. And then, as we know where the reads come from, we know what the perfect assembly would look like. And this is basically our gold standard assembly, right? Everything which is covered at least with 1x by any of the reads will end up in the gold standard. And of course, we know that this is nothing a real assembler would ever assemble, but this is what we compare the data sets to, right? And a real assembly would probably look like this. So this is even still more scattered than the gold standard. But that's a basic idea. So if we look at the marine data set, which is one of the CAMI2 challenge data sets, we have 10 different samples, 10 different profiles, basically. And for each sample, we generate 5 gigabytes of Illumina impact bio data. And this is altogether, you get something like 50 base pairs per sequencing technology. All the data is available on the website. So if you want to participate, just go to the website and you can download the data and play around with the data. So for the assembly submissions, we got well, a different mix of submissions actually. So altogether we have 12 participants with 10 different tools. I've listed the tools on the left. So this is not the same. This is not the same order as the tools here. So we are still in the process of evaluating it. So I will not reveal the real tool names. So we come up with these sort of well, arbitrary names. But we have 10 tools in total, and as you can see, some participants used the same tool and submitted results for these tools. Altogether, we got 43 submissions. And so I will focus in the rest of the talk, I will focus on the co-assembly, which means the co-assembly will pool all the short read or all the long read or even all short and long read data and assemble it basically. So how does it look like? And what we do, and for the assembly, what we do, we use the tool MetaQuast to evaluate the results. And what MetaQuast does, it just takes the context of the assembly and maps them back to the references. And then it comes up with different metrics. I will show you just a couple of these metrics because there are like lots of metrics you can look at for forever, basically. So when we look at the marine data set, there are 977 genomes in there. What we do to distinguish the genomes is we calculate the pairwise average nucleotide identity between the genomes. And we divide basically the genomes into two different groups. One group we call the common strains, which are genomes which have at least one other genome in the data sets, which has an average nucleotide identity of 95% or higher. And these are 303 of these 977 genomes. And that leaves you with 674 genomes which are unique. Unique meaning there's none other genomes which has 95% identity. And you can imagine that it's probably easy to assemble these genomes because they are pretty unique in the data set, and it's more challenging to assemble these common strains. And let's look at the results here. So this is showing, so each dot basically is showing one genome. And on the x-axis, you see the sequencing coverage of this particular genome. And on the y-axis, you see the genome fraction, meaning like how much of the genome has actually been reconstructed by this particular assembler. And what you see on the right here are the different submissions. So on the top, we have the gold standard. Here, this is the gold standard of the short reads. This is the gold standard of the long reads. And what you can see is with pretty low coverage, you basically don't get any assemblies. If you increase the coverage, you increase the fraction of the genome you can assemble. And what you would expect to see, and what we also actually see on this plot here, is that if you use longer reads, you get actually more genome recovered with lower sequencing coverage already. And if you pool these data sets, and you get these hybrid assemblies, which is even better. And compared to the first CAMI challenge, the assemblers look pretty similar now, right? There is not a single assembler which completely fails, at least not for these unique genomes, right? So all of them are very similar in the type of genomes they generate, and the only thing you notice is that there's a little shift to the right here. So meaning that this particular assembler up here is actually able to assemble more of the low coverage genomes compared to the other ones. So if you add the common strains, and these are, as I said, these are the more challenging genomes, you see that these common strains are not very well recovered in the assembly. And this is something we actually observed in the first CAMI challenge also. And apparently there's not a lot improvement because we still lose all these common genomes which have similar genomes in the data set. And even if you combine these data sets, so this is an assembler that does actually a hybrid assembly, and this is, I think, the only one that generates a hybrid assembly using the short and the long reads, you don't actually see much improvement right now. So I think there's still room for improvement for the different assemblies. And of course, you can look at all the different other metrics that MetaQuest gives you, but in the interest of time, I will just skip this. I will just give you a very short overview of the strain madness challenge. In the strain madness challenge, we have very, very closely related strains. That's why it's called strain madness. We have 100 different samples with different profiles, and we generate 2 gigabases of data for Illumina and PacBio for each of these samples. There are 409 genomes in here, and as you see, there are only 14 unique strains, and most of them are actually more than 95% identical on the nucleotide level. And if you now look at the assembly results, so in this case, we only have 7 tools and 38 submissions, and we will look at the co-assembly again for the short reads. And as I said, there are not many unique genomes, so there's not much to see here. But what you see in this plot is that basically all of the assemblers just fail to assemble these genomes, right? So this is a huge challenge. And as you can imagine, if you have lots of these very similar genomes in your metagenome, you might miss all of these in your final assembly. And with this, I will end acknowledging the core team and also Alexei Gurevich, who helped a lot using the MetaQuest and improving the MetaQuest. Okay, thank you very much.
Thank you, Alex. That was wonderful to see. We don't have any questions in the Q&A, but please, audience, type questions in there. You can do it after Alex's talk if you like. I've got one question, Alex. About the long reads. There was one assembler submission with long reads only. How did that one behave on common strains? Do we know yet?
I'm sorry, the hybrid one?
Long read only? Do we have a long read only assembly?
I would say it's not much better than compared to the short read assemblies, actually. So I think there's still a lot of work to do.
That's good. This is why we do these things. It highlights challenges